We are hurtling toward the day when climate change could be irreversible.
Rising sea levels already altering this nation's coast.
China's capital is choking in its worst pollution of the year.
5% of species will become extinct.
Sea levels rising, glaciers melting.
Okay, what is climate change? According to the NASA, climate change is a long-term change in the average weather patterns that have come to define Earth's local, regional, and global climates. These changes have a broad range of observed effects that are synonymous with the term. Climate change causes the Earth's weather to be less predictable, and in polar regions, the warming global temperatures melt the glaciers and ice sheets in an accelerated rate, and hence cause the rise of sea level all around the world.
How microbes contribute to climate change? Microbes play an important role in the production and consumption of powerful greenhouse gases, which can suffer perturbations due to anthropogenic climate change. However, the role of microbial communities is still under research, as microbes survive in diverse communities that interact with the environment and other organisms in multiple ways. Therefore, microbial activity in climate models is gaining prominence in the field of climate science. Now, we will explain how microbes can negatively impact the climate by methane production and aerosol formation. Methane production by microbes. Methane, a greenhouse gas, causes increase in temperature by trapping heat in the atmosphere. The heat trapped by a single molecule of methane is about 80% higher than carbon dioxide. Methanogens are microorganisms generating energy by producing large quantities of methane during the decomposition of organic matter in hypoxic conditions. The rise in atmospheric methane level in recent years was closely related to the increased emissions from methanogens and the reduced atmospheric methane oxidation. Ruminant animals are the main source of anthropogenic methane emissions, with a 20 to 50 times larger carbon footprint for ruminant meat production than high-protein plant-based foods. Methanobacter ruminantium, an example of methanogen undergoing anaerobic for fermentative degradation of organic compounds into organic acids and some gases, such as hydrogen and carbon dioxide. In addition to the anthropogenic methane production associated with fossil fuels, methanogens produce methane in natural and artificial anaerobic environments such as water-saturated soils or rice paddies, sediments, gastrointestinal tracts of animals, especially the ruminant animals like cattle, sheep, and goats, wastewater facilities, and biogas facilities. Methanogens can utilize hydrogen to reduce carbon dioxide into methane under strict anaerobic conditions. However, anthropogenic climate change is predicted to increase the methane emissions from rice production in a few decades due to the methanogens. Aerosol formation by microbes. Aerosols are small suspended particles in a gas that can affect cloud formation. Particularly, phytoplankton emits dimethyl sulfide, and its sulfide derivatives promote cloud condensation. The rate of aerosol influencing sunlight irradiation, precipitation, and the ways of aerosol influencing climate could take a long time to be determined. Investigations on the ways of marine phytoplankton contributes to aerosol will allow better prediction of how changing ocean conditions affect cloud formation and climate feedback. The sources and nature of marine aerosols are influenced by the combination of ocean emissions, ship exhaust, and transported continental emissions, in which ocean emissions are affected by the presence of phytoplankton. Marine aerosols consist of a complex mixture of sea salt, non-sea salt sulfates, and organic molecules that promote cloud condensation and affect the radiation balance and climate. For instance, biogenic aerosols in remote marine environments like Southern Ocean can increase the number and size of cloud droplets. They have similar impact on climate as aerosols in highly polluted areas. To add to the point, since a large number of microbes is present in the atmosphere, which is an estimate of 10 to the power of 22 microbial cells in the atmosphere, therefore, investigating and determining the ability of atmospheric microbes in growing and aggregate formation will be important for assessing their consequences on climate change.
Even though what we heard just now was all about the bad side of the microbes, but Do you know that, actually, there are certain microbes that can limit the effect of climate change? Microorganisms are very important to use greenhouse gases as energy source and build their cell. Therefore, when microbial communities and biogeochemical cycles are linked together, it is a good mechanism to solve climate change. First, we will talk about microbial communities and carbon cycles. Secondly, microbial communities and methane cycles. And lastly, microbial communities and nitrogen cycle. Firstly, microbial communities and carbon cycle. Microorganisms are part of a larger cycling of carbon that occurs on the global scale. Much of the carbon that enters the carbon cycle is carbon dioxide. This form of carbon exists as a gas in the atmosphere, but before it can be incorporated into living organisms, it must be transformed into usable organic form. The transformative process by which carbon dioxide is taken out from the atmospheric reservoir and fixes into organic substances is called carbon fixation. The well-known example of carbon fixation is photosynthesis, a process by which energy derived from sunlight is harnessed to form organic compounds. Photosynthetic algae are important microorganisms in this regard, and chemoautotrophs are mentioned. Primarily, bacteria and archaea are capable of carbon dioxide conversion into sugar form available for cell building. Some organic carbon is returned to the atmosphere as CO2 form during respiration. The rest of the organic carbon may cycle from organisms to organisms through in food chain. When an organism dies, it is decomposed by bacteria and its carbon is released into the atmosphere or the soil. CO2 dissolves in the water. At that time, algae, plants, and bacteria convert into organic carbon. In anaerobic environments, microorganisms can cycle the carbon compounds to use energy in a process known as fermentation. For example, green and purple sulfur bacteria are able to use the energy they gain from the degradations of a compound called hydrogen sulfide to degrade carbon compounds. Other bacteria such as Tuobacillus ferrocidens uses the energy gained from the removal of an electrons from iron-containing compounds to convert carbons. The anaerobic degradation of carbon is done only by microorganisms. This degradation is a collaborative effort involving numerous bacteria such as Bacteroides succinogens, Clostridium botulinum, and Centrophormonas species. These bacteria will consume atmospheric CO2 and either store it or produce biofuel and foodstuffs. The Earth's temperature will fall back to normal level, and it also prevents ocean acidification, which increases the ability of marine life to extract calcium from the water to build their shell and skeletons. Secondly, microbial communities and methane cycle. Methane is a greenhouse gas most of the time enters to atmospheres because of microbial action. Methanotrophic bacteria consume methane as their only source of energy and convert it to carbon dioxide during their digestive process. These bacteria can consume huge concentration of methane, which is helpful in reducing methane emissions from methane-producing factories and landfills. These include Methylocinus, Methylocytes, Methanomonas, Methylomonas, Methanobacter, and Methylococcus. Next, in anaerobic conditions, carbon dioxide easily changes into methane that is accomplished by methanogenic bacteria. The conversion process needs hydrogens, yield water, and energy for methanogens. To accomplish the recycling pattern, another group of methane bacteria called methanol oxidizing bacteria or methanotrophs can convert methane to carbon dioxide. In the presence of oxygen, CH4 is oxidized to CO2, yet use water and energy by methanotrophs. Methanotrophs tend to live at the boundary between aerobic and anaerobic environments. They have access to the methane produced by the anaerobic methanogenic bacteria, but also access to the oxygen needed for their conversion of the methane. Let's have a look at the overview. The bacteria that we mentioned just now consume CH4, thus reducing the methane emissions to the atmosphere. This is the fastest yet most effective way to slow the rate of warming now, as less heat travels in the atmosphere, which slows down the melting of the iceberg. Lastly, we will talk about microbial communities and nitrogen cycle. Most of the time, microorganisms push the system in order to harvest energy or to accumulate nitrogen in a form needed for their growth and development. The major transformations of nitrogen are involved through the following steps. First, nitrogen fixation. Nitrogen is in the form of a very stable molecules which is unusable by plants and animals without fixation. Therefore, two kinds of nitrogen-fixing bacteria are recognized. The first kind, the free-living bacteria, includes cyanobacteria or blue-green algae, nostoc, azotobacter, and Clostridium. The second kind comprise the mutualitis bacteria, mainly rhizobiums associated with leguminous plants. Nitrogen fixation is carried out by free-living and symbiotic microorganisms in a good manner. These bacteria have the nitrogenase enzyme that combines gaseous nitrogens with hydrogens to produce ammonia, which is converted by the bacteria into other organic compounds. Secondly, nitrification is the process ammonia transformed into nitrates by living things. Nitrates are what the organisms can absorb. The transformation of ammonia to nitrate is completed by soil-living bacteria and other nitrifying bacteria. In the primary stage of nitrification, the conversion of ammonia to nitrites is done by bacteria such as Nitrosomonas species. Other bacteria species such as Nitrobacter are responsible for the oxidation of the nitrites to nitrates. Third, assimilations. This stage indicates that the mechanisms of plants get nitrogens. Plants can obtain nitrates from soil by their root hairs. Eventually, it is used in cellular component production like amino acid, nucleic acids, and chlorophylls. In plants, they have a symbiotic relation with rhizobium. Some nitrogen is assimilated in the form of ammonium ions directly from the nodules. Other life forms also seek nitrogen through food chain structure. Ammonification is the stage of decaying. During living things are die, decomposers like fungi and bacteria turn nitrogen to ammonia. Later, it can re-enter in the normal nitrogen cycle. The process is termed as ammonification or mineralization. Many types of enzymes are involved, for example, GLN synthetase, GLU2 also glutarate aminotransferase, and GLU dehydrogenase. Denitrification. At the end of cycle, extra nitrogen molecule in the soil move off to the atmosphere. Denitrification is the reduction of nitrate back into the largely inert nitrogen gas for completing cycle. This kind of task is performed by special and unique group of bacteria like Pseudomonas and Clostridium. They use nitrate as an electron acceptor in the place of oxygen during respiration. The denitrifying bacteria use nitrate in the soil to carry out respiration and consequently produce nitrogen gas, which is inert and available to plants. The process takes place in the absence of oxygen commonly in waterlogged soils. Eventually, nitrates is converted to nitrogen gas and re-enter to atmosphere. Overview. The air and water can become less polluted due to less emission of nitrogen. Less nutrient pollution has impacted many streams, rivers, lakes, bays, and coastal waters. This will reduce environmental and human health issues and slowly revive the economies. Secondly, it also prevents algae blooms. This will restore the water quality, food resources, and habitats. It also increases the oxygen that fish and other aquatic life need to survive.
Now we shall see how climate change affects microbes. Climate change affects microbes by altering the environmental conditions, subsequently changing the adaptation of microbes to a new climate. Environmental factors such as temperature, pH, and nutrient availability modulate the microbial tolerance, persistency, and resistance, thus the increase and decrease in survival rates of microorganisms. In the interaction between climates and microbes, climate can affect microbes in multiple ways, which climates can change the temperature, pH of surroundings, precipitation, and microbial adaptation. Climate affects microbes by changing the temperature. Temperature alters cellular, physiological, ecological, and evolutionary processes that affect the survival of microorganisms. Climate change in terms of temperature is predicted to increase the rate of antibiotic resistance of some human pathogens. A research data suggested an increase of the daily minimum temperature by 10 degrees Celsius will lead to an increase in antibiotic resistance rates of Escherichia coli, Klebsiella pneumonia, and Staphylococcus aureus by 2.2 to 4.2%. Potential underlying mechanisms include increased temperature promoting horizontal gene transfer of mobile genetic elements of resistance, and increased pathogen growth rates promoting environmental persistence, carriage, and transmission. Population growth that amplifies climate change is also an important factor in contributing to the rise in resistance of microbes. Climate affects microbes by altering the pH value. Increase in greenhouse gases results in ocean acidification. Ocean acidification causes changes in the pH of the living environment of marine microorganisms. This affects their intracellular pH homeostasis. The size of microorganisms, aggregation state, metabolic activity, and the growth rate influence the capacity for pH regulation. Microbe species that are not good in regulating internal pH will be affected more. Lower pH causes bacteria and archaea to change the gene expression in ways that support cell maintenance instead of growth. Experiments has been done to prove this. In mesocosms with low phytoplankton biomass, bacteria consume more resources to pH homeostasis than bacteria in nutrient-enriched mesocosms with high phytoplankton biomass. Consequently, ocean acidification has a possibility to alter the microbial food web through changes in cellular growth efficiency, carbon cycling, and energy fluxes, with the biggest effects expected in the oligotrophic regions, which include most of the oceans. Experimental comparisons of Senichicus species growth under both present and predicted future pH concentrations show effects not only on the cyanobacteria but also on the cyanophage viruses that infect them. Climate affects microbes by changing precipitation. Fungi and bacteria in soils occupy specific niche in the ecosystems. Climate changes result in increasing and decreasing the precipitation and soil pH, will affect the abundance, diversity, and functions of the fungi and bacteria. For example, climate change of increase in aridity can reduce bacterial and fungal diversity and abundance in global dry lands. Reducing soil microbial diversity reduces the overall functional potential of microbial communities, thereby limiting their capacity to support plant growth. Increasing temperature and drought strongly affect the ability to grow crops. Climate affects microbes by changing the microbial adaptation. Climate change can increase the disease risk by altering the host and parasite acclimation, in which the microbes become accustomed to a new climate. For ectotoms such as amphibians, increasing temperature causes susceptibility to infection, possibly through perturbation of immune responses due to microbes. For example, unpredictable environmental temperature fluctuations increase the susceptibility of the Cuban tree frogs to the pathogenic chytrid fungus named Batrachochytrium dendrobatidis. The impact of increasing temperature on infections is in contrast with the decrease in growth capacity of the fungus in pure culture. This hypothesis indicates the importance of determining the effects of climate change when assessing host pathogen responses, rather than simply extrapolating from growth rate of isolated microbes in a culture medium.
Okay, so now we'll move on to our conclusion. From this video, we know that microbes cause the climate change by production of methane gas to increase the temperature and aerosols in cloud formation. Microbes limit the effect of climate change by carbon cycle, methane cycle, and nitrogen cycle. Temperature alters the survival of microbes. Ocean acidification affects their intracellular pH homeostasis, and climate change affects the precipitation and hence affects the abundance, diversity, and functions of microorganisms. Climate change affects the microbial adaptation, and investigations on the climate change allow host pathogen responses. It is to know that human activity is the main cause of global warming. In my opinion, we as human beings, the residents on the earth, need to play the most important role in preventing this climate change, and we can't blame others for this. What we need to do is to reduce the greenhouse gases production in our daily life. So let's take care of our earth as the best we can, not only for you and me, but for all of the human beings.